So the last speaker in this session is Behr. He's the director of the in vitro fertilization clinic here. Why don't you come back up around this way? That's the easiest way back.
Okay, I was watching you.
Okay. And he's worked with Renee when she was here and continued work with Tom Behr and David Camarillo, working on trying to understand early stages in development. With that, I'd like to introduce Behr.
I see what Renee's talking about, the lights. It certainly is bright up here. And good morning, everyone. It's really an honor to be here, to be amongst this distinguished faculty who's spoken before me. And as Chris mentioned, I have collaborated quite extensively with Renee, and I'm going to try put together sort of a translational story for you about the clinical work we do and how some of this research that goes on here and other places sort of interplays with the clinical story. And really, when you think about child and maternal health, the best opportunity to impact the health of the future person is when you have the gametes in a dish or under a microscope in front of you. So I'm going to share with you a story about how we interrogate the gametes and how we make future people in the in vitro fertilization practice. But before we talk about in vitro fertilization, I think it's very important to appreciate how the natural reproductive process works, and hopefully it's not going to be new to most of you in the room, but it is important to recognize that a woman is born with all the eggs she's going to have, and about every month, one egg only is matured out of approximately 1,000 that is lost every month. And as a woman ages, the likelihood of a viable egg being ovulated is reduced. So it's very important that we understand these limitations so that we can address them when a couple presents to us with a difficulty conceiving. Also, it's important to appreciate it's the blastocyst, about the day 5 to 6 embryo or 7 embryo, depending on the species. At least in the human, about the day 5 or 6 embryo is what arrives in the uterus for implantation. And up until a few decades ago, actually up until just before I came to Stanford, the standard of practice for in vitro fertilization was to transfer tubal stage embryos, the 4-cell or 8-cell embryo, to the uterus at a time prior to implantation was ready to occur. And what the major problem with this was, and still is to some degree, is that we had to transfer more embryos than we would want babies. And oftentimes we were caught short and would have multiple embryos implant beyond what we would feel is necessary or appropriate for a human. And this certainly impacts the health of the baby and the mother should a multiple pregnancy occur. So I want to talk to you about some of the ways we can improve embryo selection, alleviate some of these sort of de facto problems of multiple pregnancy and early delivery, and see how we can actually potentially improve the quality of life for the people that we are creating, not just maintain them. It's also important to appreciate that, aiding in or procreation is not a new idea. If you see here on the top of the slide, in 1790 was the first successful birth from artificial insemination. So the concept of trying to assist fertility followed really shortly after the invention of the microscope. And the little image there to the right of the text is Van Leeuwenhoek, who invented the first microscope in 1765. His impression of what a sperm would look like, this little tadpole-like structure with a little man inside. Or actually, he was close. It could be a man or a woman, because as you know, the sperm determines the sex of the embryo. I also want to move forward in history. This is a picture of Louise Brown. She was the first baby born from in vitro fertilization in July of 1978. So when you think about IVF, it's a very new profession. The oldest product of this profession is really only 38 years old. Important to remember, 2 of the 3 people in that picture have passed away, unfortunately. But the gentleman on the right, Bob Edwards, was the first IVF practitioner to win the Nobel Prize in Medicine. And unfortunately, his clinical colleague, Patrick Steptoe, was not alive to receive the award. Otherwise, he too would have been awarded that Nobel Prize. But this is how IVF works. IVF is a team sport. It works with a scientist, an embryologist like myself, and Bob Edwards, with a clinician, a practitioner, physician, gynecologist, reproductive endocrinologist like Patrick Steptoe was. And very important, the nursing team, Jean Purdy, who's in the middle, sort of to unite, coordinate all these functions. But when we're talking about IVF, what is important? The single most important sort of determining factor in the success of IVF is the patient. We can't currently change the raw materials, so what we're presented with is what we have to work with. And I've circled for you on the right-hand side there the embryo. Which embryo? Because the focus of my research for the last couple of decades has been how to identify the most viable embryo. And you heard a little bit from Renee in her talk about how we use time-lapse that I will share with you a little bit more as well, but that's certainly one opportunity that we have to help identify the viable embryo. Certainly the expertise in the lab and the program, the conditions, etc. are important, but what I want to focus on for my presentation, and as I have for my career mostly, is how to identify viable embryos. I also want to mention the Barker hypothesis. If you don't know about this, I think it's an important concept, theory, or hypothesis to be aware of. David Barker published in 1990 some work that described that, I think it's called Fetal Origins of Adult Disease, essentially describing how birth weight is associated with late onset diseases in adulthood. And I put this sort of scheme together with my focus on embryo selection. And if we took if we extrapolated back, so not from fetus to adulthood, but we took from fetus back to embryo, from embryo to fetus, how could embryo selection impact the birth weight of a child? And it could do it in several ways. It could do it by improving embryo selection so that we replace one embryo. It could do it through genetic screening so we ensure we don't have genetic malformations. We can do it by transferring embryos earlier if we could identify their viability and limit the potential imprinting defects or epigenetic effects that culturing embryos in basically Kool-Aid in a plastic culture dish could induce. So the IVF lab is very important in vitro fertilization process. As you can see here in the slides behind me, there are 7 or so steps involved in doing an IVF cycle, and these begin with stimulating many eggs to develop or to mature. As I mentioned in my first slide, normally 1 egg matures every month out of about 1,000 that are recruited. And one of the goals of good IVF stimulation would be to try to rescue more than 1 out of those 1,000 that are being recruited so that we have more candidates to make embryos for selection. So I'm not going to go through all of these in detail, but you can see there are many opportunities that we can interrogate or question or investigate the gametes, the embryos, prior to them being implanted. And this includes doing genetic testing on them. I'm going to end my talk by talking about pre-implantation genetic screening or diagnoses, which essentially allows us to do an amnio on an embryo before it's implanted. So to avoid essentially the implantation of undesirable embryos or pregnancies. So how does IVF begin? I mentioned recruiting many eggs, but once the eggs have been recruited, we need to get them in our hands or in our incubators before they're ovulated. So this is a very sort of acutely or specifically timed process where we use transvaginal ultrasound to access or visualize the ovaries that you can see in the bottom panel. Those black circles are actually fluid-filled follicles that are about 20 millimeters in diameter that contain an egg. A human egg is about 100 microns in size, so you can't see that with your naked eye, but essentially the fluid from the follicles are aspirated and then passed off to the embryology laboratory, which is a highly specialized lab. I'll show you some pictures of our laboratory at the end as well, where the conditions are maintained for keeping gametes and embryos viable outside the body. Embryos don't have an immune system, they don't have liver, they don't have lungs, they have no way, they don't have kidneys, so we have to essentially provide the perfect clean environment for them so they can thrive. This is what an egg looks like. You were introduced to the days by Renee, and day 0 is what we note as the day the eggs come out. And the first really opportunity to interrogate the viability of an egg, which I'll state is not clinically routine clinical practice yet, would be to analyze these cells that surround the egg. These are called corona radiata, or granulosa cells. They're modified granulosa cells. These are the cells that actually are endocrine organ. They actually produce steroid hormones during the stimulation process that feed back to the hypothalamus in the brain to essentially result in the normal process in 1 egg maturing. So we interfere with this through our superovulation regime and can actually now, and I'm working with Steve Artundi's lab currently in looking at the actually TERT expression. It's involved in telomerase lengthening in these cumulus cells. And it turns out in animal models there is an association between egg viability and the cumulus cells. These cells play also a critical role in preparing the sperm for fertilization. So in the natural condition or in traditional in vitro fertilization, where we add sperm to eggs, if we remove those cells from the eggs, the sperm wouldn't be able to fertilize. So they play several roles in reproduction, and we feel that these could be an index of viability of the egg from the very beginning. If we denude the egg, as we would to look at these cells, you can see that the egg is presented in a variety of maturational states. So starting off with a germinal vesicle, that is basically a diploid cell, a primordial germ cell, and it goes through meiosis under control of the hypothalamus and pituitary to achieve a mature metaphase 2 oocyte, which is the stage that the oocyte arrests in. The egg, if the egg never sees a sperm, it's not activated, it will stay in this stage viable for about 24 hours before it will become atretic and be non-viable. So again, the timing, we heard about timing. Timing is critical, not only for assessing viability, but also for allowing normal development to take place. So some other work, and again, I'm not going to show you data, I'm going to show you some more pictures and be more descriptive to be the translational portion of the session. But to show you some pictures of some incredible work I'm doing with Nader Pramond at UC Santa Cruz, where we're using a nanosensor that can actually aspirate femtoliters of fluid from cells, actually intra-organelle. So we can go into the mitochondria inside a cell and measure the pH. It is absolutely incredible, but this is some work we're doing. A 3PN is an abnormally fertilized oocyte, which is not viable, and it is a tremendous opportunity to sort of investigate egg potential because it has no viable potential to make a person, but it has a very great research potential. Well, what about the sperm? The sperm obviously is a critical structure. You need one sperm and you need one egg. And the sperm are made through a very intricate process called spermatogenesis, where unlike an egg, men make sperm their whole life. So we are fortunate that we are deployed with primordial germ cells that are self-renewing. And we start off with a diploid primordial germ cell. And again, unlike an egg, we make 4 mature daughter cells for mature sperm. But some key things happen during the formation of a sperm cell. One is a sperm has no cytoplasm. So a sperm loses all its cytoplasm during spermatogenesis, and it also changes its size. It dramatically shrinks, and what allows this to occur is sperm has a unique packaging of DNA out of any cell in our body. Sperm's DNA, as you'll see in the bottom right there in the blue, sperm's DNA are wrapped around protamines, which allow the nucleus to be highly coiled and much, much smaller than an egg nucleus. And I'm not going to show you pictures of that today for the sake of time, but I want you to appreciate that the packaging of the DNA and the cytoplasmic removal has a dramatic impact on the sperm's viability and their morphology. And when we do a semen analysis on a man, we see these kinds of morphologies, and these are due to, sort of aberrations in the spermatogenic process. In fact, men are, extremely inefficient in this process. For example, for us to be considered normal, we only have to have 4% of our sperm looking normal. So we can have 96% abnormal sperm and still be considered normal with respect to morphology. And this is highly problematic. Compared to a bull, for example, a bull has 96% normal sperm versus 96% abnormal sperm. So it's quite amazing that we actually exist with this degree of abnormalities. And we do have to take these consultations seriously, because it does take 2 to tango, and we need just as many sperm as eggs to make the process work. So to continue on this opportunity for interrogation, doing some more work with this nanosensor technology, we can actually go into an individual sperm, and we've actually sequenced individual sperm from RNA. So actually being able to take RNA out of the sperm, keep it viable, and sequence the sperm. And this you can see circled here or behind me is the sperm after it's been sampled. This is not a video, so you can't appreciate it maintains its motility. And we're in the process now of sort of testing sperm to ensure that they indeed can do all the things they need to do after the aspiration. So what we do mostly nowadays to achieve fertilization is a process called intracytoplasmic sperm injection. Many men present with below 4% morphology or low concentration. So what you're looking at now is a sperm loaded into a pipette that is being advanced into the egg to achieve the sort of displacement or deposition of the sperm into the oocyte. But before we actually deposit the sperm, we aspirate the cytoplasm back. So you'll see if you keep your eye on the ball or eye on the sperm, you'll notice it moves almost out of frame, and that is sort of the sign that the egg membrane has been broken, and we can now deposit the sperm directly into the cytoplasm. And this is sort of mechanical fertilization. You can see we've denuded the egg. And it is another opportunity that I sort of discovered for viability assessment. And I noticed that when we inject the eggs, some of them are hard and some of them are soft, sort of like Goldilocks, you know, too hard or too soft or just right. And working with, as Chris mentioned, with bioengineering, we've developed an assay that can assess embryo viability through their viscoelastic properties that I'll show you in a minute. But if the injection is successful, we will see this, which are 2 PNs, 2 pronuclei. You heard about the male and female pronuclei. This is a fertilized egg. And I want to show you a video of how we can assess the fertilized egg's viability. And that 3-second video that you saw there, where you see the invagination of the zona pellucida, the eggshell, and the cytoplasm of the 2PN, allows us, when we apply an algorithm, to predict viability in a way that's better than anything we've done so far, comparable, if not better, to the time lapse, which takes approximately 48 hours. We call this embryo mechanical phenotype. And I want to show you, we've only done, we published a Nature Nature Communications paper, came out in February, and we started a clinical trial. We've only done 22 patients so far, but really we just received a Coulter grant, so we're looking forward to taking this a little bit further, getting more data. But you can see quite clearly here, on limited numbers, so there are no statistics, I apologize, that the blue bars are higher than the red bars, showing that sort of the area under the curve, the correlation between these mechanical properties and day 3 morphology, comparing day 3 morphology predicting blastocyst or pregnancy rate, are significantly improved. So we're highly optimistic that this will be yet another tool that we can apply to our embryos and help our patients, or help us make better decisions to assist our patients in achieving a pregnancy. So the next time we look at embryos is 2 days later. We're now on day 3, and we hope to have an 8-cell embryo, something that looks like this. But the dilemma is, as I told you, we make lots. And how do we choose which embryo to implant? And there's no one in this room, even collectively no one in this room, and there are a lot of smart people here I know, could identify which embryo to choose or which embryo to implant. It's not possible with the information provided. So, and actually, to give you an appreciation for those of you who don't work with embryos, this is an 8-cell embryo sitting on the point of a pin. So you can appreciate how small 100 microns is, and this is what we're dealing with on a daily basis. We all looked like this, by the way, 3 days after fertilization. But again, the dilemma is which embryo to transfer. This is a picture from, a decade or so ago when we were routinely transferring more than one embryo. And the problem is this underscores the problem. We have 6 embryos on one patient on the right, 1 embryo from a patient on the left. They were both transferred, and guess what? They both resulted in a singleton pregnancy. So we don't do this on the right anymore. We culture embryos to the blastocyst stage, which really allows us to make better selections. But morphology is limiting. What you see is not always what you get. And I want to point out to you here that these nuns are sitting on bar stools. These are not their legs. And it's just to sort of underscore that, depending on your mood, depending on your perception or your position, you may see things differently than your neighbor. And we need to do better than this in the science. And to that end, I want to show you now a sort of fast forward about 3 years from Renee's pictures on the time lapse to a more clinical version of the time lapse movie. This is 18 embryos growing in wells now that are indexed, so we know which embryo is which. And we've changed the circles to be different colors for each division. And you can appreciate in this picture that not all embryos are dividing at the same rate. And it turns out this timing is highly informative, and I'm very proud to say that Stanford IVF Lab, starting May 2nd, will be the only lab in the country that is doing this on 100% of their patients. So we have really taken sort of the bench-to-bedside story and taken it to reality where, as basic scientists and clinicians, we've made a collaboration, we tested it, we published it, tested it, and a company was started, and essentially the product is now in our hands for our patients to take advantage of. And as you can see, this high and low refers to what was in a prior video, viable and non-viable, the likelihood of implantation based on the sort of morphokinetic parameters. The human blastocyst is sort of the ultimate stage that we hope to achieve in culture, and most of our patients' embryos are grown to this stage for transfer or definitely for cryopreservation. We have a very successful cryopreservation program for supernumerary embryos, and we use a technique called vitrification that essentially was developed for egg freezing, or egg preservation, which we now offer as well, that we apply to blastocysts, which is actually spectacular. This is a day 6 hatching blastocyst or day 5 hatching blastocyst, and it is at this stage that we really like to perform our pre-implantation genetic screening. I want to sort of put the whole story of embryos to blastocyst in a movie for you, and this is published by a colleague of mine, Yaz Mio, in Japan. But this is just a very nice and the first high-resolution video of human cleavage showing the compaction stage at the morula, which you'll see right there, and then the expansion of the blastocyst, the stretching of the zona pellucida to the point that the embryo is able to hatch at about 1 o'clock, you'll see in a minute, before it's ready to implant in the endometrium. And again, this is a process that we visualize now quite routinely in the lab, and every single one of us in this room, or every single person on this planet, had to do this before their mother could be pregnant. So in the last few minutes, I want to sort of wrap up with the ultimate form of embryo selection or embryo screening, and this is what we call PGD or PGS, pre-implantation genetic screening or pre-implantation genetic diagnosis. And this really, as you can see here in the bullets on the screen, is a process that we use for couples undergoing or individuals undergoing in vitro fertilization, where we have access to the oocytes or the embryos where we can screen for chromosomal number problems like Down syndrome or sex selection, or single gene defects like Tay-Sachs, cystic fibrosis, etc. So essentially any gene that we know the sequence for, we can sort of fish for or find, spell check in the embryos. We're not doing any gene therapy or any gene editing on human embryos. We are still doing screening or selection, elimination of genetically abnormal or unfavorable traits, but I can imagine, and you all can imagine, that the future probably has these gene editing technologies in the future. So I have 2 videos I'm going to show you after this, but I wanted to show you these 3 pictures, which represent the 3 stages that we can actually interrogate the fertilized oocyte. On the bottom left in the blue, those are polar bodies that are being removed. I didn't have time to sort of review meiosis with you, but you need to appreciate that why an egg doesn't make, or a primordial oogonium doesn't make 4 equal daughter cells is because it goes through a process of meiosis where it makes 1 viable daughter cells and 3 polar bodies. So the polar bodies is somewhat looking in sort of your garbage can to figure out what you had for dinner last night, but it can be quite accurate in looking what's not inside the egg, and this technique can be used for maternally inherited conditions only, clearly because it only has maternal DNA. The most popular time to do genetic screening is on day 3 worldwide, even though I contributed to the first publication, actually myself and Ruth Lighty, one of my clinical colleagues, published the first paper on trophectoderm biopsy, which is on the top right, on frozen thawed embryos a decade or so ago. So although the day 3 biopsy has value and has merit, I think from the work we heard from Magda this morning, and for reasons why we actually went down the path of trying to develop the time lapse, we weren't convinced that those cells were all the same on day 3. And even though they look the same, I think we had concerns, which has now borne out to be true, that these cells molecularly had some fate that we were unaware of. And should we remove a cell that was due to be, had fated to be in the inner cell mass, we could compromise the embryo beyond the level we would want to. So now doing trophectoderm biopsy as a routine, as you'll see in this following slide. I'm going to have these 2 videos playing in parallel, just for the sake of time, and you can appreciate the hatching blastocyst on the right and the day 3 embryo on the left. What's going on here is we're using a laser to breach the zona pellucida to get access to that one cell that's by the hole. And on the right, we're slowly aligning the pipette to aspirate 5 or 6 trophectoderm cells into the pipette so that we can use the laser to cut them off. So it's much like giving the cells or the embryo a haircut. We feel that the day 5 biopsy is a better choice because we can take 5 cells instead of 1 cell. And we can take 5 cells from a 100-cell embryo instead of 1 cell from an 8-cell embryo. So these although comparable in their potential to diagnose the embryo, the day 5 biopsy is a more robust approach and probably less detrimental to the embryo. So you can see the nucleus, if you look closely on the day 3 on the blastomere, it looks like a little crater inside the cell, and that cell would be removed and sent to a molecular genetics lab, while the remaining embryo would either be frozen or allowed to be placed in culture. And what's going on the right is that we're actually using the laser to cut or dislodge the cells from their neighbor so that we can have 5 or 6 cells in the pipette that would then be dispensed into a PCR tube essentially and sent off to the molecular lab for the analysis. The next generation sequencing technology and array CGH technology is becoming faster by the day, cheaper by the day, and I think it's not going to be long before it's probably going to be irresponsible to do IVF without doing genetic screening. And then the people on our faculty, Hank Greeley specifically, and I'm sure many of you have heard of him talk, or maybe read his book that has just come out, or maybe not yet, that talks about sort of the end of sex, and it's going to be irresponsible in the future to have children without doing IVF so that you can ensure they'll be healthy and normal. So with that, I'm going to try, I've got 50 seconds or so. I'm going to end by telling you that this whole process culminates with an embryo transfer, with the transfer of either, as I mentioned here, an 8-cell embryo on day 3, or ideally a blastocyst, and hopefully make healthier people. I'm going to end right there. Thank you very much.
Do you have some questions for Barry? So Barry, you've been working with optics and mechanical tools and a variety of ways of assessing these embryos. What's in the future? Will we be using optical tweezers to pull and push on these embryos to try to figure out what their mechanical properties are?
I think the future is bright. I think we are going to be taking advantage of technology. I believe the future will be robotic IVF. With the advent of microfluidics, with all these cell sensing technologies, the ability to sample cells, I think we're going to be testing gametes and using a tested sperm and a tested egg to make a viable embryo, and not make 20 embryos and be left with 6 in the tank and twins with mom who are not going to have more kids, and then left with ethical dilemmas and moral dilemmas what to do with these leftover embryos. So I think it's a little bit sort of Gattaca-ish to some people, but I think with responsible people like people here at Stanford in this room, our goal is to make healthier people better lives, not designer babies.
Another question?
So I'm Zulfi Bhutta from Sick Kids Toronto and Aga Khan University. Fascinating stuff. You started off by alluding to the Barker's hypothesis. And I wondered, in all of this in vitro work, is there anything that allows you to predict SGA, or being born small for gestational age, from any of the morphology studies or characteristics that you have of the oocytes?
I think the answer is not yet. I mean, one thing that we can do that is sort of 50,000 foot is make sure that you have a singleton pregnancy, because certainly, and I did have another couple of slides that I chose not to show here at the end before my thank you, which shows the risks of having twins that most people who come to in our waiting room, if you polled, would say, we want twins, we want twins, we want twins. So to answer your question, easily, eliminate twins and you will get to normal birth weights or closer. Small for gestational age, I believe the MFM's in the room, and maybe Dr. El Sayed can talk about some of the work that they're doing that will potentially be able to predict this from CVSs and other things, but not yet from the egg or the embryo, to my knowledge. But I hope to get there. That would be a terrific sort of added value to our diagnostics.
Thank you.
Thank you, Barry.
Thank you. Thank you.
Let's thank all the speakers in this session.